These are their faces. I'll see you later, guys. Okay, so at the moment there's little bits of paper being handed out, so these we'll come back to a little bit later. But today the focus is on inflammation and asbestos. So last year, if you did patho, I would have covered inflammation a little bit with you. If you've not done it, it's not a problem, but you should have some notes at least you can look back at from last year as well. But in terms of inflammation, I'm going to talk to you a lot today about how it's a bad thing. So it's the same as oxidative stress. I'll bang on about how kind of detrimental it is, but inflammation really is a defensive kind of physiological response. So you need to activate inflammatory cells and stimulate inflammation to protect yourself from the harm of pathogens, for example, or chemicals that might cause you harm. So don't forget that when we go through kind of the bad side of inflammation today, that ultimately you want inflammation. You want to be able to protect yourself from these potential pathogens. So I've showed you this before, or last year. So this on the left-hand side is a histology picture of your lungs. So you can see here, these are all the air spaces, alveoli. On the right-hand side, you have a picture of a lung that has quite a strong inflammatory response. It's been activated. So the scale is slightly different. So this kind of represents this red circle, one of these alveoli. But you can see this, the lung, the air spaces are packed full of inflammatory cells. So you can imagine that the function of the lung is probably compromised in this circumstance. So this is quite an extreme representation of an inflammatory response, but actually inflammation can stimulate something that is damaging and harmful to your body, as well as a protective response. So normally what happens during inflammation, as you saw on that previous slide, is that you get a huge amount of inflammatory cells that are recruited into the site where you've been exposed to a foreign material. This recruitment of the cells can cause damage to the normal healthy cells within that organ, and this is because by their nature they're inherently kind of toxic. They're trying to remove foreign material. In order to do that, they tend to release things like reactive oxygen species. So we spoke about this during oxidative stress. So reactive oxygen species are harmful. So if the immune cells release these, it could help to damage microorganisms, for example. They might also release proteolytic enzymes to break down proteins that are part of the harmful pathogen. So these are needed for defence, but they're often non-specific. They'll end up targeting healthy tissue as well. So you can imagine if healthy cells are exposed to reactive oxygen species or exposed to these enzymes which break down proteins, it might compromise their function. So whilst they were released for kind of a beneficial purpose, you might not always see only the pathogen being targeted. So this is when inflammation becomes harmful. You know, if you have a persistent, we call it often chronic inflammatory response, something that's long lasting, you start to tip the balance more towards the harmful side of things rather than it being protective. So what kind of cells contribute to inflammation? You remember the names of?
Yes.
Say again?
Mast cell.
Yep, a mast cell. So we spoke quite a lot about mast cells in terms of asthma last year if you did patho, but they tend to be involved in kind of allergic type responses. What did you say?
Eosinophil.
Yep, eosinophils. So again, asthma, we spoke about them quite a lot in terms of allergic responses to allergens. Any others? Yep, so neutrophils, type of white blood cell. Say again? Selected. So those are like adhesion molecules, so it's not necessarily a cell, but a protein molecule. So they're definitely involved in inflammation, but they wouldn't be classified as a cell. Any others? Yeah, so don't forget that it's not just immune cells that contribute to inflammation, non-immune cells contribute. So I don't know if you all heard, but he said epithelial cells. So these could be epithelial cells in your gut, in your skin, in your lung, for example, and they could be activated to secrete cytokines that then stimulate inflammatory responses. Anything else? Dendritic. Yep, dendritic. So antigen-presenting cells, and generally, so we have dendritic cells or macrophages, for example. They're all there as part of your innate immune system to help protect you. So you can already see that we're listing quite a few different types of cells. So if you've done Lizzie's immunology course, you might have come across more of these types of cells. So we'll focus quite a lot on macrophages, monocytes, neutrophils, in lymphocytes, mainly in this course, but there is quite a large, diverse range of different immune cells that can contribute to inflammatory responses. I can't go through the function of each and every one of these, so just be aware that they exist. And also for the non-immune cells, we discussed epithelial cells earlier and how they have this barrier function in the body, but they can also contribute to inflammation. But it's also things like liver cells, skin cells, fibroblasts. There's a huge different range of non-immune cells that can also stimulate an inflammatory response. So remember, I won't cover all of these. I'll tend to mention in relation to particular substances, specific immune cells and how they contribute, but if you've done Lizzie's course in immunology and you've got information you can bring in from that, then that's also acceptable if I ever ask about inflammation in this course or in the exam. Okay, so in terms of where these immune cells come from, it depends on what type of cell they are normally. So within certain organs, you might have immune cells that are already resident. So it means they're already located there and they're kind of ready to respond to foreign material when your body is exposed to them. So for example, in the lung, you might have alveolar macrophages. In your liver, you have something called Kupffer cells. So these are macrophages that are found within the liver. You have dendritic cells located in many different organ types. Within the skin, for example, they're called these Langerhans cells. So these cells are already there. So they're part of your innate immune system to help protect you from potential pathogens. You can also have immune cells that are recruited to the site where you need them, so the site of injury or the site of infection, for example, and they can come from a variety of sources, but they mainly come from blood, for example. So neutrophils are the kind of predominant white blood cell that circulates within your blood, and they can leave the blood and enter, for example, the lung or any target site that you need them, or they can also come from lymph nodes. So this tends to be more lymphocytes that you'd associate with being recruited from a lymph node. So some cells will already be in an organ and can respond immediately. Others, you can imagine it takes a little bit of time for these cells to be recruited. You have to have cytokines released which attract these cells and cause them to move from their site of, or move from the blood or the lymph nodes. In terms of immune cells and how we investigate them in the lab, knowing a little bit about what they look like is often quite helpful for us when we perform a toxicology study. So here I've kind of listed the main kind of families of white blood cells, and you can see they all have different morphology. Monocytes and macrophages tend to have like an egg-like structure, but a nucleus within. They tend to be one of the biggest ones, quite a lot of cytoplasm surrounding it. Whereas if you see a lymphocyte, they tend to be much smaller, and the majority of the cell is its nucleus. For the neutrophils, you can see it's got quite a lobed nucleus, probably about 3 or 4 different lobes, and there's lots of granules inside them. The eosinophil tends to kind of have a kind of ring-shaped nucleus. When we look at them in the microscope, they tend to look quite red once we've stained them because they're quite acidic granules found within them. And basophils, we won't discuss these much in the course, but these are filled with inflammatory mediators and often packaged into these vesicles, and you can see them kind of within that cartoon. So, as I said, when we want to do a study in the lab, we want to try and quantify exactly what immune cells are being recruited, because it tells us something about what the potential implications might be for your health. So if you have eosinophils, for example, it's quite an allergic response that might have been stimulated. Later on we'll discuss kind of the time course of inflammation, but what generally happens is you have neutrophils being recruited first, and then you start to see other types of immune cells. So by looking at what immune cells are present and at what times, it can tell us something about if that immune response is quite acute or short-lasting, or whether we expect it to carry on over time. So what we do in the lab is we take samples and we stain them. So we use dyes, basically, that allow us to identify the nucleus, or nuclei of the cells, and this and other stains allow us to look at the cytoplasm. And by looking at the structure of these different cells, it allows us to identify what populations we have present. When we do this, we call it a differential cell count. So it just means we're looking at the different types of white blood cells and what proportions they're present within our sample of interest. So when it comes to doing kind of animal or human studies, there's 2 main sources of the samples that we get to perform that work. So for animal studies, it tends to be a lavage or some sort that is performed. So you can do this in humans as well, but the bronchoalveolar lavage is related to the lungs. So you take saline, the animal is dead by this point, but you wash out the lungs with the saline, and that allows you to collect all the immune cells that have been recruited to the lungs following exposure to your test substance. You can also lavage things like your peritoneum, so you can wash out all the cells that are found there, but it is more, it's a kind of more general inflammatory response you're looking to there. You probably have to inject something into that area and then look at the response. Within blood, when people have go to hospital, for example, and you're looking at the populations of different immune cells in the blood, it can tell you, if you've got more or less neutrophils circulating, it might tell you if more are being produced or if there's not enough of them to deal with a certain type of infection. So this is where we get our samples from, and then we take those samples and we stain them to try and visualise the immune cells and then count them, the different proportions of them. So what I've given you, or what hopefully you've got in front of you, or one between two, is kind of a picture or a field of view that you might see down the microscope. So all I want you to do is take a look at this and try and identify what immune cells you have. And then at the end we'll compare to see if our proportions are similar. So what to do is count the total number of cells and then count the number of macrophages, or all I've got in terms of these counts, like the more you did it, the more you would practice and you'd get more familiar with the cells, but it is quite difficult to sometimes tell what a cell type is. So anything that's kind of large circle, you can see some cytoplasm, tends to be a monocyte or macrophage, right? They tend to kind of look like eyes. The neutrophils, it might be clearer on the screen than it was on your handouts, but you can see there's lots of different lobes here. So I think most problems came from distinguishing a neutrophil from an eosinophil maybe, and when you were going through it. The eosinophils tend to have a bit of a pinkish colour to them. The stain tends to really emphasise the cytoplasm. So these two, I'd say, are the ones that cause me the grip. This could be an eosinophil, for example, this one, or this one, you can't really see the lobes. But hopefully, even if you didn't get the exact same numbers as what I did, you kind of got a similarity in that there was the most neutrophils were present, and then it was followed by these. So two different researchers doing these counts wouldn't necessarily get the exact same numbers of cells, but you would hope that the pattern of the response would be the same, that you'd highlight that there was more neutrophils over anything else. Someone asked me about, in real life in the lab, what you should do. So this is called like one field of view. What you would do is move to another part of the slide and count a different part of that. Then go to another part of the slide and count that. So you're getting different fields of view and counting maybe 200 or 300 different cells in order to hopefully get something that's representative of the whole slide or sample rather than just one little portion of it. So yeah, this isn't necessarily the most common thing that people perform in the lab, but if you do any sort of animal work, then it is something that is used to quantify the inflammatory response, and that this is just an example of a study where they performed that type of work. So you can see they had different types of chemicals. This just represents the total number of immune cells, but here you can see eosinophils, lymphocytes, neutrophils, and depending on the substance you give those animals, so this is in lungs of rats, you get different proportions of inflammatory cells. So I can't really point that high, but if you see the top one here, this BC, this is vehicle control. So this is just a normal lung, and hopefully what you can see is that it's only really macrophages that are there. So within a lung normally, all you expect to see is macrophages, because they're the only immune cells that should be resident within the lungs. As you go through the different treatments that the animals have received, you can see this one here, for example, it still tends to be quite a lot of macrophages, so you're not seeing much of inflammatory response. With this one, you can start to see some neutrophils, for example. This one, it's more, you see how they kind of look like donuts in this, like a kind of circular structures with eosinophils. You can start to see the different proportion of cell types. So you can do this type of work to compare different chemicals and see if they vary in the type of inflammatory response that they might stimulate. But the take-home message from this is that mainly in the lung you only have macrophages, there shouldn't really be any other cell types, and that the proportion of those cell types can be different. You can see this one's almost all neutrophils or a couple of eosinophils. So it's a good way of quantifying that inflammation. you can now get machines that will do this for you in terms of, it doesn't just rely on sitting in front of a microscope and doing it. It will suck up the sample into a tube and count the different types of immune cells based on how they scatter light. But at least this, you can take an image, it's trustworthy, you can see the cells as well as counting them. Okay, so as well as looking at inflammatory cells, you can look at cytokines. So remember, the cytokines are proteins. I've provided some examples here of cytokines that come up in this course, but they'll have different names and they'll all activate different types of cells or stimulate and recruit different types of cells. But they're really responsible for telling either cells to come to a site where they're needed, where there might be an injury or a site infection, or they are responsible for activating those cells. So they might bring in a neutrophil and then activate it so it starts to release reactive oxygen species, for example. And these cytokines are involved in different parts of immune responses. When you do a toxicology study, you can look at cytokine expression. So you look at the gene, the level of these genes, and if they're upregulated or downregulated, or you look at the proteins themselves, whether or not the production's been increased. So you could look in those samples that we discussed before, like blood, bronchoalveolar lavage, or you could take the lung and look in the lung tissue and see what levels of these cytokines you have. Something you can do in vivo and in vitro. So the differential cell counts you can only do in vivo, because in vitro is, you can have a flow of cells, but it would be really difficult to mimic an inflammatory response or the recruitment of the cells in vitro. But in vitro, you can look at cytokine productions as kind of an indicator that something might have the potential to stimulate inflammation. So what we can do together maybe is discuss how you might look at an immune response in vivo. So I quite often ask this type of question, not this exact one, in exams, give you a scenario, ask you to describe an inflammatory response and then say how you'd investigate it. So there's a similar process that you can use when answering that question. So you have to think about what model you use, how you expose it, well, the administration route, the times that you look at a response, and then the responses. So you can imagine this could, in vitro it's slightly different in terms of the route, but you could imagine you could do this for, so it's in vivo or in vitro. So this is asking for an in vivo response, so what model might you use if you were, so it's asking about lung inflammation to investigate in vivo. So what animal might you use?
Mouse.
Yep. What else could you use? Could potentially use a human. Yeah, so you could use monkey, anything. I'd say mouse or rat is generally the two, if I was you, I would give the example of, because they're the most commonly used. Ethically, it's probably more acceptable to use these. If you said you wanted to use a pig or a monkey, you would probably have to add a statement of, there's already been testing done in another type of species to prove that that it's not just that day one you'd go in and put it in a human, or go in and put it in a monkey or a pig. Okay, so assume you use a rat or a mouse, so how would you expose them to your substance? Yeah, so inhalation. So remember, the reason you do that is because we've asked for a pulmonary response. So by inhaling it, it's going directly into the lungs. Any other way that you could expose the lungs?
Can you have it syringed into the lungs?
Yeah, so I quite often abbreviate it as the IT, but it's the intratracheal instillation. So remember, that is kind of like a subretinal. Think of it that way. You're just depositing the material directly into the lungs. Okay, so these are the two most common routes. If you suggested like injecting into the blood, you'd have to think how your lung would be exposed. So you'd have to check that your substance got there. So it's not to say if you did suggest that, you wouldn't get marks in the exam, but you just need to justify it. Not the same thing twice. Okay, so the next thing is the time point. So when might you look at inflammation?
Just before to establish a baseline.
Potentially, but I've written something here. So everything you should have a control animal for. So your control if you're raising your animals appropriately, there should be no inflammatory response. By doing an intratracheal installation, you might cause some damage, but generally not. So if you imagine, so you wouldn't need a 0, let's say a before, but what times might you look at inflammation? So I'd say, we'll discuss this in a minute, but you can have an acute response that might be maybe within 24 hours. So it's a reasonable kind of time to suggest. You might have a week, you might have months, or you could have years potentially. So it might depend. This hasn't asked you, you don't know anything about the substance being administered, but if you knew it was something that you were exposed to daily for months on end in an occupational environment, you might suggest a longer exposure, a longer kind of experimental duration. But I'd say 24 hours. It's always good to suggest more than one. You say you look at an acute response after a short amount of time, and then maybe something after a week or a month to see if the inflammatory response resolves. The control, so remember this would be your mice just breathing in normal air, or having an intratracheal installation but not having the substance actually injected. You might want, so we'll call it a negative control, maybe a positive control. You might want to give some animals an inflammatory response to check you can detect it. The problem with that is that ethically it might not be so acceptable because you're giving an inflammatory response which might be painful, for example. And remember, you've got vehicle controls. What responses might you look at?
Physical change in behaviour or...
Yep, so you could do the behaviour stuff, but in terms of if you were looking at inflammation in particular, what might you focus on?
Sale equipment.
Second?
Sale equipment. Inflammation sale equipment.
Yes, I thought you said tail recruitment, and I was like, oh, I've not heard of this. Yeah, cell recruitment. So exactly what we've just done then, right? So you might want to take a lavage of the lungs, wash out the immune cells, and then count the different types of cell populations that you have, right? So that would be looking at the cells, and then if you did different times, you could maybe see how those cells changed over time. Anything else?
Swelling and temperature?
Yep, so you could look at things like temperature, because things like a fever, for example, can be a sign of inflammation. Swelling, I guess you probably have to do more histology and look at organ structure. You're unlikely to maybe see physical swelling in the lung because of an response in the lungs. But in terms of inflammation, if you look at the cells, but what brings the cells there? Yeah. So the cytokines, you could look at the bowel fluid or the tissue itself, so the lungs. So then, I've suggested, we've suggested these, but then you could suggest how you perform that work. So you do a differential cell count maybe, or histology, or this you could use, hopefully you've all heard of ELISAs, for example, or PCR. So it's always good to suggest how you might investigate something as well. So hopefully I've summarised just the ones relating to inflammation. I still think these are completely relevant in terms of responses, but because this is focusing on inflammation, you might want to focus more on the kind of inflammatory endpoints. But you can try this out yourself when you're revising. Imagine I ask you, if it happened in the gut, what might you do? Or the skin's slightly different because we've got different ways of looking at responses. But see if you can list the different types of responses you should measure. In terms of, I only want to cover this briefly because it should be things you know, and we've already done it there. But acute responses tend to happen within hours of exposure to material. It tends to happen because neutrophils accumulate, so those are the first type of cells you expect to increase in number. You want your neutrophil response to resolve, so generally what happens is those neutrophils die. So when I say here this timely removal of neutrophils, those neutrophils should undergo apoptosis. So they've been brought into the site where they're required, they've accumulated, and then they should die, normally within a day or so, and it's macrophages that come along and then clear those dead neutrophils. So we'll come back to neutrophils later in this course, but these should be the first type of cells that come in with kind of a day, a week maybe. And it's only if you start to see things like lymphocytes and lots of macrophages increasing in numbers that then you might be concerned that you're starting to stimulate a more chronic, persistent response. This fibrosis might be a relatively new term to some of you. So fibrosis happens when your body's trying to repair itself. So I told you that the inflammation starts to damage healthy cells. Your body tries to replace those cells, but it doesn't necessarily replace them with the same cell type. So, you know, you've all cut your finger and you see kind of a red scar forming. It's because fibroblasts have been activated and they're depositing collagen and extracellular matrix. They're trying to replace that kind of damaged tissue. So you might not replace the cells that you've lost or damaged with the exact same type of cell, and that means that you might compromise the function of that organ. So fibrosis, you need it to clear and repair the tissue, but you might lose some of the function in the organ that's been affected. So this kind of figure represents that process, and that you have like exposure to something foreign. It could be a pathogen, a chemical, whatever. You should see neutrophils. This is normal, it's physiological, and you expect that. Then you might start to see macrophages, lymphocytes, and you know, if you have a kind of physiological, normal, healthy response, then that should resolve and you start to repair any damaged tissue. If it's really long lasting, you start to go more towards the kind of pathological side where you might promote disease development and stimulate fibrosis. So it is a fine balance between what is physiological and what do you need in order to help remove this potential threat, and then where does it become pathological. It's kind of it's not necessarily the most understood process. Okay, so I want to focus in the last wee bit on macrophages, and in particular how they contribute to asbestos toxicity. So I'll always say macrophage and monocyte and use them interchangeably, but actually monocytes are what circulate in blood. They're made in the bone marrow, they're released into blood, and then they'll migrate into a tissue where they'll differentiate into a macrophage. These macrophages are found pretty much in every organ of your body. As I said, they're part of the innate immune system, there to protect you from potential harm. And they've got several different ways in which they can help remove that threat. So they can secrete cytokines, so they're trying to bring in other immune cells. So in the lung, if you're exposed to something foreign, so macrophage will recognise it, and then they'll release cytokines that bring in neutrophils, for example. They can phagocytose foreign material, so it just means they internalise it. And hopefully break it down within the lysosomes within them. So they can, this is meant to represent like bacteria, could be neutrophils that have died, it could be a chemical substance. And they can also have this respiratory burst where they release lots of reactive oxygen species. So macrophages are really good at helping to defend your body. You can see none of these are necessarily specific in terms of, they don't necessarily, they might know it's a bacteria, but they wouldn't know what bacteria it is. They just know it's foreign to your body. This phagocytosis, I've just put this process up as a reminder hopefully, but if you have a foreign material, it's taken up into kind of a membrane-bound structure within the cell, that fuses with a lysosome to form a phagolysosome, and the acidic nature of a lysosome, and it contains enzymes that can be quite destructive, should help break down that foreign material to remove it. Quite often we want to investigate whether or not a macrophage will interact with foreign material. We can use light microscopy to do this. So that picture up the top there is macrophages exposed to carbon nanotubes. So carbon is black, so we can see it, but not all chemicals or substances you'll be able to see just using light microscopy. So we have to rely on things like fluorescent microscopy. So up the top right, I think that's just some particles that are about 200 nanometers in size, but you can get fluorescent E. coli, for example, or bacteria that you could visualise within your cells. You can also do electron microscopy, so you could look and see your substances inside your cell. This is carbon nanotubes that are kind of projecting outside the cell, but the electron microscopy gives you much more higher resolution images. So for asbestos, I'll cover it a bit today, and then Fiona will do more later in the week, but hopefully you've heard of asbestos, and most people know it's quite a bad thing for your health. But asbestos comes in many different forms and it's not equally toxic. So it's naturally occurring, and you can get man-made fibres that look like asbestos, but people used to mine this material and use it in different products. And up the top here you can kind of see it's like javelins. It's really long, it's thin, it's fibre-like structure. The reason people used it, it was a really good insulator, so they used it in buildings, they used it in lots of different products where they needed heat resistance. And lots of people were exposed to asbestos in an occupational environment. So you imagine if they were putting insulation into a home or using it in construction, quite a lot of it was breathed in during that process. I've showed you this picture before, I think, so you know that this particular type of cancer, this mesothelioma, that's associated with asbestos exposure. This is the tumour here developing around the lung. It's a really severe cancer. It's got quite a poor prognosis, which means we don't have a cure. If people are diagnosed with this disease, it tends to happen at a point where surgically you can't remove it. There's no good kind of drug cures for this. So this meant because we know it's a harmful substance that asbestos was banned. We can't use it. But we do know that asbestos toxicity is driven by its structure. So we know if the fibres are really long and biopersistent and thin, that they tend to be much more toxic than shorter, non-biopersistent fibres. So this is a good structure-activity relationship. We can relate the structure of these materials to how pathogenic they might be. And within the lungs, when they're inhaled, macrophages are what's key to their pathogenicity. The activation of macrophages is what promotes the development of cancer to this material. So in terms of the lung, you can see at the top right here, you have the lung structure, and then the pleural cavity surrounds the lungs. So this allows your lungs to move in the body when they inflate, allows them to stay inflated, for example. It's a really important part of the structure of your lung. But what happens is these fibres are so long and thin, they deposit in this outer region of the lung. So they've got to get past, like the alveoli, for example, they've got to get through all the thin tubes, the airways in your lung, and reach this kind of cavity that surrounds it. Once they're in that cavity, they interact with macrophages, which are there for protection, and the cavity is lined by mesothelial cells. So these are like an epithelial cell. They're lining this cavity, and what happens is that the macrophages themselves are about, let's say, 10 to 15 microns in diameter. If the asbestos fibre is along with that, the macrophages really struggle to clear them. Here you can see, these aren't necessarily asbestos, it's just fibres that we've worked with in the lab, but these are fibres, all these kind of stick-like structures, and to see these are cells. So the cells aren't big enough to phagocytose the whole material because they're much longer than the cells. So you can see quite a lot of cells might accumulate. This one's kind of been pierced along it. It's really struggling to internalise them because they're too long. The fact that they're biopersistent as well, so that term just means that your body can't break them down. They're going to stay that length. They can't chop it down into smaller portions that could be more easily ingested by the macrophages. So you can see this causes the macrophage some distress. It's wanting to clear them, but it can't. So we call this frustrated phagocytosis. It just means the fibres are too long. They can't be internalised by the cell. And the fibres are biopersistent. So the macrophage will try and release reactive oxygen species and enzymes to break down the fibre to make it shorter, but the fibre can't be broken down. It's made of material that your body can't break down, so then that's stimulating inflammation. The macrophage wants other immune cells to help, so it starts to release cytokines, which brings in nutrients, for example. You start to see proteases, reactive oxygen species released and causing a damaging response. So overall, this kind of inflammation and oxidative stress is why you get cancer, because the mesothelial cells are exposed to high levels of reactive oxygen species and a lot of inflammation, and then that promotes cancer development within the mesothelium. So we know quite a lot about why asbestos is harmful. We know that if, and by short I mean 10 microns or so, we know if fibres are short, then it's not a problem. So when people find out this information, they started to make synthetic asbestos that was much shorter, that could be cleared from the lungs. If you have the long fibres, if they're non-biopersistent, it's not a problem because your body can break them down and clear them. It's only when they're persistent that you start to see a toxic effect because your body can't break them down to something shorter. Carbon nanotubes is kind of the focus of your assessment, and the reason why we're concerned about carbon nanotubes is because they look like asbestos in terms of their structure. So people are excited about them because you can potentially use them within a kind of endless number of different applications. So industry really want to exploit these materials. They're really strong and light, so they want to use them in cars, they want to use them in things like tennis rackets, golf clubs all have them to make them lighter. But because they look like asbestos, people are a bit concerned that we might have a similar adverse health effect emerging to these materials. So people took what we know about asbestos and applied it to carbon nanotubes to see if they elicited a similar response. So what people do is they take their carbon nanotubes and their asbestos and they see what they look like. So they use electron microscopy, and you see there's kind of these long fibres, these structures here, look kind of like the pictures of asbestos I showed. Sometimes it looks more, we call these like more entangled or spaghetti-like. They're not these long, thin structures. So you can imagine if you just looked at this, you might think, if these are over 10 microns in length, we might have something damaging. Because these ones might be more easily ingested by macrophage, maybe we're less concerned about them. So you can look at them and try and decide if they're toxic using microscopy. This study here is kind of similar to what you've got in your assessment, but it's an in vivo study. These are long carbon nanotubes. These are kind of entangled or short ones. This is a long, a short asbestos. This is long asbestos. This is carbon, so it's carbon nanotubes are made of carbon, but they're fibre-like in structure. These are kind of carbon spheres. They're spherical in nature. So they want to see if carbon nanotubes are toxic, is it because they're made of carbon, or is it because of their structure? So it's a kind of control within the experiment. So in this study, they injected them into the peritoneal cavity. So this is kind of the abdomen, and then 24 hours and 7 days later, they sacrificed the animals and counted the inflammatory cells that accumulated within that cavity. And what you can see is that this is the neutrophils here. It's a kind of the indicator of inflammation. These ones are all short or tangled, so this is asbestos, these are carbon nanotubes. They're not eliciting an inflammatory response. Anything right at this line is a long fibre, so long asbestos, so long carbon nanotubes, and they're inducing quite a strong inflammatory response. So this kind of tells us that if something is long, then it's more likely to be toxic than something that's short. And so this kind of corresponds to what we saw with asbestos. They also took images of the cells that they took from the peritoneal cavity, and they could see little fibres or the tangled structures within the macrophages when they gave them something long. This is what they saw. They kind of saw them pierced within the structure of the carbon nanotube. So again, it might suggest you have some sort of frustrated phagocytosis, that the cells can't clear these fibre materials. So this I won't ask you, but overall, this suggests that, again, length is important, that you're stimulating inflammation in the lungs, and it's probably because of the fibre structure, that they're long and biopersistent. In terms of in vivo studies, obviously they use animals, so ethically it might not be so acceptable. So we're trying to move away from them. So people have also looked at them in vitro. So what cell might you use if you were doing an in vitro experiment for a carbon nanotube?
Macrophages.
Yeah, so macrophages is the one I would suggest you focus on, but you could use a mesothelial cell because they're the ones that ultimately develop into a cancer cell. And I think Fiona will give you an example of that. But remember, macrophages, they're phagocytic, they're involved in your immune system, they're there to clear them, and we know that for asbestos, macrophages play a really prominent role within the pathology of the disease. So this study, conducted a few years ago now, again it was really like the animal study I just showed you. They took a panel of different carbon nanotubes. So some were quite long and thin here, like they're like 50 microns in length. This one is like 10 microns, but they kind of interact with each other, and then this one's just a big mess. They're all kind of agglomerated or aggregating together. So this is to show you something in toxicology. It's quite difficult. Each one of these on the label would say carbon nanotube, but actually they might all elicit a different response. When it comes to regulating these materials and deciding whether or not they're safe, we might not necessarily be able to group them all within the same thing and say all carbon nanotubes are unsafe. They might all elicit something slightly different in terms of their toxicity. So this time they took images of the interactions of the materials of the cells. So this is your control, kind of normal, happy-looking cells. This was the really long fibres. I don't know how easy it is for you to see, but you can see they're kind of stretched out. You can see that there's big fibres or long fibres contained within the cell, and that they don't look the same as the control. These ones, you can kind of see the carbon materials inside the cell. They kind of look more like the control. So this might suggest that this one on the top right was causing a frustrated phagocytosis. So they investigated this using more biochemical-like endpoints. They looked at superoxide production. So this is like a respiratory burst. It shows you if the macrophages are releasing reactive oxygen species, which would suggest to you that they're kind of they can't degrade the material and they can't internalise it, so they're trying to break it down by releasing reactive species. And again, it was only kind of the longer fibres that could do this. The more entangled one didn't elicit a response. And then TNF-alpha is a pro-inflammatory cytokine. So again, the longer fibres are inducing a response, whilst the shorter entangled ones aren't. So this in vitro experiment maybe gave some evidence that the shape of the fibres was also important to their toxicity. You notice this is long fibres of asbestos, and it's not as toxic as the carbon nanotube. So this was only done at one time point at 2 concentrations. You probably need more asbestos to elicit the same response as a carbon nanotube. And that in itself is quite telling in that it's more toxic than something that we already know is extremely toxic. So again, we have evidence that the structure is important, that if it's long and thin, it's more likely to induce a toxic response than something that is shorter, and that macrophages play a critical role. And then, in terms of this in vivo versus in vitro, hopefully I've showed you that you can kind of come to the same conclusion on the toxicity of carbon nanotubes if you've performed an in vitro experiment than if you might have performed an in vivo one. So you're not looking at exactly the same responses, but it's kind of similar. So in both circumstances, you saw the uptake of the materials and that the macrophages struggled to internalise the long fibres. So you got that information if you did an in vivo or an in vitro experiment. You could look at a pro-inflammatory response, so you could look at cell accumulation in vivo, or you could look at cytokines in vitro. You also saw that the length was important. It tended to increase the toxicity the longer that they got. So, there's quite a lot of scope for saying that maybe we can't get rid of animal testing completely, but maybe you would start in vitro and see what response you got before deciding if you'd made the most toxic carbon nanotube, maybe you wouldn't want to use it in all your products, and an in vitro study might tell you that without having to progress to an in vivo study. One thing I want you to think about, Fiona will go over this with you later this week, but you might have seen maybe it was an exam question from last year, I can't remember, but I asked you to kind of design a testing strategy. So based on what you know about carbon nanotube, if you were given a new sample of a carbon nanotube, how would you test if it was toxic? So think about how I've told you it's toxic, what it kind of stimulates in cells, and then you can think about what different levels of testing you might perform. So would you go straight into an animal, or would you do in vitro? If you went in vitro, what would you do? So we'll talk about it maybe as part of like a revision session for this course, but these type of activities are mainly for you in your own time to help you with revision, rather than for me to give you all the answers. Is there any questions? So yeah, I'd recommend you go to Fiona's lecture because it will help you with your assessment as well. And if you can just take all the wee bits of paper and put them in the bin or recycle.